compartment, but not in others, we discussed on the previous slide. Again, due to availability of substrate or the presence of a specific enzyme. So if we don't have something, it's going to be really hard to make that pathway happen. If we do have it, it makes it a lot easier. And compartmentalization prevents futile cycles. We don't want to waste energy. We burn up to, I think she said, 6 grams of glucose an hour in our brains. So we really want to make sure that we're not wasting energy, otherwise our brain's going to starve, and so will all of our other organs. So this is why, unlike bacteria, we have organs that are specifically designed to perform functions. So the liver's main purpose is to maintain blood glucose. And our adipose tissue is meant for storage of fatty acids. So when we're out of energy, we can go to these fatty acid storages and get what we need. Muscles also store glucose, but they can't export it. Liver is actually the only organ that can send glucose to other organs. So muscle keeps its glucose for itself. The brain also does the same thing, but unlike some of the other organs, the brain can only work off of glucose. She mentioned that there's other carbohydrate or sugar sources that other tissues can get energy from, but the brain can only get energy from glucose. So Dr. Harrison talked briefly about how major things that we eat are broken down. So the first one that she talked about were carbohydrates. And we have macronutrients and micronutrients. There's more information on this in your notes, but I just wanted to touch on it briefly. Macronutrients are the things that we need a lot of, like protein, and micronutrients are things that we need but not as much of, like vitamins and minerals. So then she talked about carbohydrates. And again, carbohydrates are a type of sugar, and so sugar and carbohydrate are often used kind of interchangeably. And when we talk about sugar, we're talking about glucose and things like that. And ultimately, we haven't gotten to this pathway, but glucose will get converted to maltose and dextrose. So that's their fate. Proteins are broken down as they pass through the body. So they're initially broken down just by the pH in your stomach. And the pH actually activates this enzyme called pepsin in its active form. It's called pepsin. And this also helps break specific peptide bonds. And as protein passes through the digestive pathway, other enzymes become activated. For example, trypsinogen becomes activated to trypsin, and this enzyme then activates these 3 remaining enzymes. So they kind of build off of each other to help digest food. And so fats are emulsified in the intestine, and they eventually get formed into these micelles, which contain triacylglycerols, fatty acids, and monoacylglycerols. And so we'll talk more about fats later in the course, I believe. So eventually they get hydrolyzed to, lipoprotein lipase hydrolyzes these TAGs to fatty acids so we can store energy. So lastly, she talked about vitamins. And these can actually get absorbed directly by the intestines. They don't need to get broken down or converted to something else. And this is good because we can take them up directly. But sometimes it can be a little bad because some of them are fat soluble, which means that they don't get excreted. And so you can actually get a buildup if you take too many of these fat soluble vitamins. It's pretty hard to do that, but something to be aware of. So finally, we have all the information we need to kind of jump into this gigantic pathway. And we spent last Thursday talking about this part highlighted in yellow. And we spent yesterday talking about this part here, which again, I'm not going to be able to go through today, so hopefully Gina can go through that for the next discussion. So this top part is how we convert glucose to glycogen. And glycogen is the storage form of glucose. If we don't need glucose right away, we want to store it so that we can use it for later. And there's a series of processes to get glucose into the form of glycogen. And so this is called glycogen synthesis or glycogenesis. There's a couple names for it. But basically, we're making glycogen. And this is the storage form of glucose. The reason we need to convert glucose to something totally different is because glucose is water soluble. So if we tried to store it, it would just get redissolved and sent back out of the cell. So we need to get it into a format so that it actually stays in the cell where we need it, especially in something like muscle, where we're going to need it when we need to use our muscles. And glycogen is just these long chains of glucose. So this is an individual glucose molecule, and you can see that it's just these long chains built together. There's two ends. There's non-reducing ends. So these are kind of the branch points. And then there's a reducing end, which is the very base of a glycogen molecule. And there's two different types of linkages that hold glycogen together. I don't have a good picture of the long thing, but we get these long chains, and these are held together by alpha-1,4 linkages. And they're named as such because it's carbon 1 from, sorry, I always do this backwards. It's carbon 1 from one glucose molecule connected to the fourth carbon of another molecule. And you can see that they're numbered up here. So carbon 1 would be connected to a fourth one over here, and this fourth one would be connected to C1 of another glucose molecule. And that's how we get these linear chains. But it's energetically favorable to actually make these branches. And so we have these additional connections where C6 is connected to the C1 of another glucose. So they're slightly different. They serve the same purpose of holding glycogen together, but they are slightly different. And so ultimately what needs to happen before glucose can get added to a growing glycogen strand is that glucose needs to be converted to UDP glucose. It's kind of the big picture. Once it's in this format, we can then add it to glycogen. But it has to go through a series of steps in order for that to happen. So glucose, with the addition of ATP and this hexokinase enzyme, again, kinase means that it's adding a phosphate, which makes sense because it's taking the phosphate from this ATP. And we get glucose 6-phosphate. I have it on the worksheet, but it wasn't really in her slides. She vaguely mentioned it once. There is also this glucose 6 then goes to glucose 1, and there's an intermediate between those two called glucose 1,6-bisphosphate. She sort of mentioned it. It's not totally in the notes. I wouldn't worry about it, but I just wanted you to at least hear it so that you're vaguely familiar, in case you come across it, where that fits into this whole pathway. So glucose 1,6-bisphosphate is the intermediate between these two enzymes. So glucose 6-phosphate is again converted to glucose 1-phosphate with that intermediate, and this is done by phosphoglutamutase. Glucose 1-phosphate with the addition of UTP, so we have ATP up here and UTP down here, with this enzyme is then converted to UDP glucose. And then lastly, UDP glucose can be added to this growing glycogen strand by this enzyme, glycogen synthase. So the name makes sense, glycogen synthase, because it's synthesizing glycogen. And lastly, there's this enzyme, which has the easiest name to remember, branching enzyme, and it just makes the glycogen branches. So once a glycogen strand gets long enough, it can then transfer 6 to 7 glucose residues from the end of one strand to another strand. I'm not entirely sure how it knows exactly where to cleave and where to add. I'm guessing it's some kind of allosteric recognition, but I'm not entirely sure. And this last step here is irreversible. And then once we have glycogen, if we need to burn it, we need to remove these glucose residues. And so the enzyme that helps catalyze that reaction is phosphorylase. And so this helps remove one of those glucose residues, and it actually converts it back to glucose 1-phosphate. So, yeah?
Can I ask you what's the intermediate again?
Glucose 1,6-bisphosphate. It makes sense, because we're starting at glucose 6. That phosphate is getting moved to the 1 carbon, and in between it's kind of on both. I don't know exactly how it's on both, if there's 2 or if there's 1. Again, I wouldn't worry about it much, but she did mention it, and so I wanted you guys to know roughly where it fits in. We're also going to talk about like fructose 1,6-bisphosphate, which is different than glucose 1,6-bisphosphate, so I just wanted to mention it. I hope I don't confuse you guys. So glycogen can't just be made from nothing. It's got to have something to start off of. And the thing that it starts off of is glycogenin. And this is a dimer with 2 tyrosine residues, and it can just self-add these glucose molecules to itself. And so this is glycogenin. and it auto-catalyzes, which means that there's no other enzyme involved in this. It can just add glucose residues to itself. And once they get to be long enough, then glucosynthase, the enzyme that synthesizes glycogen, can come in and add additional glucose molecules. And then lastly, branching enzyme, when this gets long enough, it'll cleave the 1,4 linkage, and it'll create a 1,6 linkage and make these branching enzymes, or these branches. And so again, just to emphasize, the reason it's really important to get glucose away from just being glucose is because glucose 6-phosphate can't leave the cell, whereas glucose can actually leave the cell. Again, there are certain tissues, there are certain organs, like the muscle and brain, that don't share their glucose. So glucose here can leave the cell, and glucose from other cells could leave, but muscle holds on to its glucose. But in the glucose 6-phosphate, it's trapped in a cell, and so it's committed to being stored at that point. So this is kind of the whole process kind of put together. So this would be glycogenesis, and this would be glycogenolysis. So glycogenolysis is when we're actually removing glucose residues to use them again, and then they get shuttled into the remainder pathway that we'll talk about, that we talked about yesterday, actually. So glycogenin would have self-catalyzed, and we've got the beginning of the glycogen strand here. And glycogen synthase will come in and continue adding glucose residues. Branching enzyme will make more branches, and this will keep going, I don't know how long, but it makes a really big molecule. Eventually, we're going to need these glucose residues back. And so it gets debranched, and these individual residues are removed, and they're converted back to glucose 1-phosphate. And so there's hormones that help regulate this. And in the liver, the hormone that's helping regulate is glucagon. And as we mentioned in this slide, in this section here, we have these two enzymes that are doing opposite things. One is making glycogen, and one is breaking down glycogen. And we don't want them on at the same time, because it would be kind of counterintuitive to be making glucose and breaking it down at the exact same time. So we need to make sure that they're not on at the same time. And that's the role of glucagon. So the liver responds to glucagon, and this leads to the phosphorylation of phosphorylase and glycogen synthase. And this has opposite effects on these two enzymes. So phosphorylase, when it gets phosphorylated, becomes activated. And glycogen synthase, when it gets phosphorylated, becomes inactivated. So this is an example of how a covalent modification can have two completely opposite effects on an enzyme. So when phosphorylase is activated, again, this is breaking down those glycogen molecules to get our glucose back. And this makes sense because this hormone is released when blood glucose is low. So we need glucose, so we need to be breaking down glycogen. So this enzyme needs to be activated. Well, if we need glucose, we don't want to be storing glucose. And so we want to turn off glycogen synthase. And so this stops glycogen synthesis. So it kind of makes sense. You just have to, it's a little confusing to remember that there are these two rules, but if you remember that they're doing opposite things, it makes sense that they can only one of them can be on. Muscles do not have glucagon receptors, but they are sensitive to epinephrine, which is also called adrenaline. And this activates phosphorylase when we need glycogen broken down into glucose. So lastly, Dr. Harrison talked about carbo loading. And if you guys have ever played sports, you've probably had a big pasta dinner right before an event. And someone finally explained to me in college that most people actually carbo load incorrectly. So the trick is that you actually have to deplete your carbohydrate supply before you have your big pasta dinner. So you eat a really high protein diet to deplete those carbohydrate stores. And then you have your big pasta dinner, and your muscle cells, because they've been depleted of this glycogen, they actually synthesize up to 100% more glycogen. So I've always had just a big pasta dinner. I've never taken it like 5 days before a big event. So I thought that was really interesting. Okay, so that's kind of all that I had to review from last week. So I've got a worksheet to help remember what all of these different steps are and what all the different enzymes involved are. So I've got this up to kind of help us go through it. So we can go through it together.